Hi, good day. Let us learn about transcription and translation. Proteins are made on ribosomes in the cytoplasm of the cell. Yet, DNA, which contains the genetic code, is found only in the nucleus. How is information from the genetic code brought to the ribosomes for protein synthesis? Scientists began to understand how proteins are made when a second kind of nucleic acid, RNA or ribonucleic acid, was discovered. Like DNA, RNA is a nucleic acid. However, RNA structures differs from DNA structure in three ways. First, RNA is usually composed of a single strand of nucleotides, rather than a double strand as in DNA. RNA also contains a different type of sugar molecule, ribose, instead of deoxyribose. Like DNA, RNA also contains the nitrogen bases, but rather than thymine, RNA contains a similar base called uracil. Today, we know that RNA is the form in which information moves from DNA in the nucleus to the ribosomes in the cytoplasm. Enzymes make an RNA copy of a DNA strand in a process called transcription. The process of transcription is similar to the process of DNA replication. The main difference is that the process results in the formation of one single-stranded RNA molecule, rather than one new double-stranded molecule of DNA. This RNA copy that carries information from DNA out into the cytoplasm of the cell is called messenger RNA or mRNA. Messenger RNA carries the information for making a protein chain to the ribosomes, where proteins are synthesized. Some portion of DNA code for the RNA that makes up ribosomes. This RNA is called ribosomal RNA or rRNA. Recently, scientists have shown that rRNA helps to produce enzymes needed to bond amino acids together during protein synthesis. Let us see this figure for you to understand the process of transcription. First, the process of transcription begins as enzymes unzip the molecule of DNA, just as they do during DNA replication. As the DNA molecule unzips, free RNA nucleotides pair with complementary DNA nucleotides on one of the DNA strands. Thus, if a sequence of codons on the DNA strands were AGC, TAA, CCG, the sequence of codons on the RNA strand would be UCG, AUU, and GGC. And when the process of base pairing is completed, the mRNA molecules breaks away as the DNA strands rejoin. The mRNA leaves the nucleus and enters the cytoplasm. Let us have the process of translation. When mRNA is synthesized, it carries a complementary copy of the DNA code for a protein chain. How is mRNA language used to synthesize a sequence of amino acids? The process of converting the information in a sequence of nitrogen bases in mRNA into a sequence of amino acids that make a protein is known as translation. Translations which occurs on ribosomes involves a third kind of RNA. If proteins are to be built, the 20 different amino acids dissolved in the cytoplasm must be brought to the ribosomes. This is the role of transfer RNA. Transfer RNA or tRNA brings amino acids to the ribosomes so they can be assembled into proteins. How does the cell know which tRNA molecules carry the proper amino acids? The answer again involves base pairing. Correct translation of the code depends upon joining of each mRNA codon with the anticodons of the proper tRNA molecules. This figure shows how the image in an mRNA molecule is translated into a protein. The end result of translation is the formation of the large variety of proteins that make up the structure of organisms and help them to function. This is the simplest process of translation. As translation begins, the first codon of the mRNA strand attaches to a ribosome. Then tRNA molecule, each carrying a specific amino acid, approach the ribosome. When the tRNA anticodon pairs with the mRNA codon, the two molecules join and the tRNA molecules remains there. Often, the first codon on mRNA is AUG, which codes for the amino acid methionine. AUG signals the start of protein synthesis. When this signal is given, the mRNA slides along the ribosome for the next codon. Again, a new tRNA molecule carrying an amino acid will pair with the mRNA codon. When the first and second amino acids are in place, an enzyme joins them by forming a peptide bond between them. Lastly, as the process continues, a chain of amino acids is formed until the ribosome reaches a stop codon on the mRNA strand. Now that you know, proteins are made by bonding together of amino acids during translation of the mRNA copy of the DNA code. Can you guess how many types of proteins can be made in this way? The fact is, an almost infinite variety of proteins can be made from the same 20 amino acids. Remember, although there are only 20 amino acids, they can occur in any order and in any number. It is the sequence of amino acids in a protein that determines the characteristics of that protein. If this is your first time watching my video, make sure you hit the subscriber button. Thank you for watching.